kakor su gärna så väldigt Du skjønte hva jeg mente.
Hei. Her er det.
Now I can see you. Right. Okay, you're getting fewer and fewer, which means it's getting earlier and later in the season. But I'll take the names just in case and all that stuff. But you have to deliver your introductions at 1 o'clock today, right? Or noon. Okay. And how's it going? Because basically, if you want some workshopping on it, we'll do it now. And if you want some answers on the results chapter, we'll do it now. So this is the question. Literally, how's it going? What do you need to get things done properly now? I have all the hard copies of the results chapters here, and I know you probably went yesterday when you got them back. Yeah, I know. But you will never again get as much feedback as you do in this course. And the feedback is going exactly on the strictly scientifically correct format of submitting it. And granted, you will find crap in the literature, but please, we don't want to teach you how to get away with crap. We want to teach you how to submit good quality standard science and analyses and a logical report. So that means that your introduction is also going to be good quality and scientifically valid and logically put together. There was at least one group that had a little problem with the logically put together. And the introduction is notorious, especially when you're not quite sure what your results say. But it's been a while since you submitted your results, so you should have been thinking about it by now. You should have an idea of what your story is. Do you want, right, and you're all looking at me like, right, okay, so I'm not sure if you're all comfortable with what you got, or you barely know where to start.
But how does the introduction relate with the results?
Right. Okay.
Because basically you write the introduction first.
Yeah. No, actually, when you are writing your thesis.
Yeah, you can come back later on, but first,
okay. The introduction is what is read first. It is not written first. And what is written first is what you have done. You've done the materials and methods, that's what's written first. Then you do the results, that's what's written second. And that is the backbone of your report, those results. Morning. Okay. So the results, even though you wrote it second, is where everything comes from. Okay. When you're looking at your results, and this was one of my comments yesterday, you are building up the blocks of a story that nobody has told before in a way that nobody has been able to see it before. Okay. And that means the only facts you have to go on are your own results. And if your own results are pointing in this way and your introduction goes this way, well, there's no point. You're not. It will be scientifically incorrect, and it's also illogical. So you have to look at your results and see what they actually say. And that means, for example, I don't know, Gudmund, you guys have a fairly clear view. Aren't you Lille Lungegaard?
Yeah.
Yeah. Okay. And your results are pretty interesting with relation to the way the ecosystem is going and a change that has recently happened. Okay? And so your introduction, give us an idea of what your introduction could be.
So we look specifically at the parameters for water quality that we're looking at because we have an ecosystem for fish, so we're adding that to our introduction, what is good quality for fish.
Mm-hmm.
And then we also felt we need
probably also telling a little bit about why you chose, or why the
Yeah.
parameters of E. coli, phosphorus, nitrate, and so on have been chosen in order to
Yeah.
analyze.
Related to the water quality or related to.
Right. Yeah. So that's one way of getting into it. Now they have a specific story, and it's pretty clear from their results. But let's see. Hannah, how about Yana? Sorry. What about your results? What did they say?
It said that the water quality was fairly good
Okay.
and that the biodiversity could be better.
Yeah.
And we were hoping to get the poor oxygen concentration in the sediments.
Oh yeah, you're real close.
Yeah.
Yeah.
So we have contacted the biogeology department,
Mm-hmm.
and we're hoping to get those results as well.
Yeah.
Because then we'll be
It will go nicely into the
Yeah.
to the discussion. Yeah.
So in the introduction we have talked about kind of the river mussel as its historical participant, and also what has been done in Hackles River
Mm-hmm.
before, what they are trying to do, and what the demands are for reintroduction of the river mussel.
Right.
So, and it seems like it's going good, but we need the oxygen core.
Yeah. The
Sediments.
pore sediment. Yeah.
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 Hopefully we'll get it.
Okay. Yeah. So you see now there's elements of the story. They have Linanungagon that has gone through several different interventions to try and make it a nice pond downtown with water. They have Haukos River, where the endangered freshwater pearl mussels are going to be hopefully reintroduced, but the environment isn't good enough yet, and there's a whole lot of tactics there. But one of the things and so that's the way you start to build the story. And but one of the things all of you have to do is link why these samples were taken. Okay. Fit. I mean, basically, it means why do TCB? Why do TCB? Why do the abiotic? Why do phosphorus? Whatever else you did, and why do biodiversity? And it might be just one sentence, or it might actually end up being a clue for the way that you are interpreting your data. But in your introduction, you give us why you did those. Because your introduction has to end with a question that is answered by the materials and methods. So that you lead to a question in the introduction through your story that says you're going to be examining the environment for example, the suitability of returning the mussels. Some of the parameters, the TCB will give you the bacteria in the water and the influence of animal and human waste products. The biodiversity will give you how well the animals in general live in that area, and so on and so on, okay? So you have to use this and explain to us in the introduction, very briefly, why these parameters, why these variables are the ones we also looked at in order to answer the question. There may be other variables that could have been used to answer the question better, but that's life. And in some cases, like Haukos, they're going to go back and get something extra because they discovered, yeah, it's really quite important. It's also right expensive and all that stuff, so it's like on a volunteer basis from BioGeo. But that's really quite good. Yes?
Basically, I was just thinking about the results, because what we did and what we get might be different. So I was just thinking, if we are going to build the introduction based on the results, so
okay, but
we might get something. Yeah. But we basically build from the what we did, the materials and methods.
The questions, yeah, you
But
build the...
if we are going to start from the results,
Okay,
that was what...
Right.
Because we might get another story.
yeah, but
But
in
we-
fact you might,
Yeah.
yeah.
So
Okay, but you're
that's what I was thinking, how
Right.
we're going to...
okay.
But
Your
maybe
starting
then-
point, yeah, your starting point is the question that Babin Kommuna asked.
Yeah.
Okay, and you went out there to answer that. In your results, surprises happen. Like in one place, there was a stream that people suspected, or that the Kommuna suspected was the source of bacterial pollution, and it was clean. You could drink from it. So there's that, and so there's other sources of pollution that have to be acting there, and that means that you can use the introduction to say, to sort of build up the framework on which you are systematically testing places of pollution source, so that you apply it at least once there, and in the discussion you come back again and you can say, okay, this method showed that such and such was not the pollution source. We recommend that such and such be carried out in these other places, which we suspect are in fact closer to the original, to the source of the pollution that we see at this water. Okay, so you build up elements. You know what your results are, and hopefully you understand the details in there. And you build up your introduction based on the question that Babin Kommuna posed originally, and the answer that you now know you actually have and have to focus on, okay? Because that's it. You're focusing on something. Remember, the introduction goes like this. So it's broad, that's the question. And down here, it's the methodology to answer the question. And then you go this way through M&M and results. And then discussion goes backwards. So you're focusing here, and you're coming back out here. But that little link means that there's a link between there, all the way through here, and then to the beginning of discussion. Okay? So you have to keep the reader focused there. And that means when you're writing the discussion, you're narrowing down and narrowing down, going from the broad site to smaller and smaller factors. But those smaller and smaller factors are in part determined by what you know is special in your own results. Okay, and I know that this, when I see your introductions eventually, I'll be seeing something that is out of context from the other chapters that you have written. So I'll only be able to evaluate the introductory chapter on its own merit, without a link to the results. But when it comes to the final version, it's that link all the way through that's going to make a big difference, and that you've been following it and thinking logically through. How do you feel about that? Okay. This also means, for example, if you have this broad question at the beginning of introduction, and then you go on about biodiversity in general, and then you go on about bacteria in general, we're not getting narrower and narrower. We're also not finding out what else has been, what other places in the world are like your site. What other places in the world have had your kind of question. So we want the literature study in there, absolutely. And if possible, also the reference values that you'll have to be taking into consideration. So the reference values for TCB, the reference values for phosphorus and nitrate if you got it, and oxygen saturation, that kind of thing. So, and that might only be half a sentence, but it'd be nice to have it in there. Because those are the bases, that's what you're comparing with. Okay? The introduction gives the groundwork, stuff that's already been done. The reference values are already there. The question is already there. Previous work is already there. And then the discussion, that's where new information does not come in, not all the stuff about already there. In the discussion, then you're starting to put your results in connection with what had been done before. You guys all look like seriously worried. Do you want to, okay, because I want to help you here, but I'm not quite sure where to start. And since you're basically all ready to submit today, is that correct? Or have you done it? Hands in the air who's ready to submit today, sort of. Okay,
We're
right.
submitting to the TA today.
Right,
is it?
right, yeah. Okay. But just so you know, I mean, I don't have to hold on, I don't have to keep you here for 2 hours if what you have to do is still work. But I'll show you, I'll just bring up, because that compendium that we put together is actually, hate this. That compendium we put together does give you the basic layout, believe it or not. And it is pretty straightforward. So hang on a sec. There, this, oh, sorry. There. This one here, so under file storage, is how to write a thesis, right? And of course you've all memorized this, right? Okay. So there's the chapters. This is the way that it is read. It's not the way it's written, okay? But the introduction, page 10. Oops. And remember there, page 2 says, start collecting literature. So I hope you've already started that. Okay, notice that here. The introduction should give a satisfactory and logically structured background for the problem without going into unnecessary detail. The problem is described and a precise question formulated. Okay? And there. You present the problem. You demonstrate you're familiar with the relevant literature. The literature counts a lot. And you mention the method that was used. The main points are to answer, why is this an interesting research subject? And how did you perform the experiment or the field study or whatever? Okay? That's what you're answering. And this is always what the peer reviews look at. And when you submit your report, I'm using the same 21 questions that you applied to the articles that you critically read. It's almost not rocket science. It's just plain convention and logic. But it's really difficult to do it when you're in the middle of it. It's really difficult to see it. It's like, I can't read what I wrote because I see what I meant and not what I said, if you understand. And I'm sure quite a lot of you have had that way. So that, so it says up there, present in a concise manner the general aspects of the field study, including the most important references. The general aspects. That means you already now, because of your own results, know what the general aspects were. You wouldn't have been able to write this first. You wouldn't have been able to write this the day after we gave you the fieldwork assignments. Now you can. What are the general aspects here? And when you present the problem, oh yeah, see that middle part? No pointer. In that paragraph it says, so present. In the middle part it says, also keep in mind that completely new information shouldn't pop up in the discussion. Give us, the readers, what we need to know what we should know before we start the reading of your story. Okay, so something important does not turn up in discussion, as in, oops, we forgot to mention this special characteristic that influenced everything else. You mention it here in the introduction. So your reference values are here, and the mention of previous studies are here, and so on. But not specifically yet, and not in relation to your results, obviously. Okay? And when you present the problem, done in such a way that the reader can easily understand the historical content of the work and why it's important. That's why literature is really important. That's why previous reports, what you know, the history of Lillehammer, history of Hagos, wonderful stuff to know. Wonderful stuff to know. And actually quite interesting for the reader as well. And for example, which site did you have? I've forgotten again.
The Kalvträvika.
Kalvträvika. Oh yeah, okay. And what was special about that?
Basically, the question was to investigate the streams affecting the bay.
Right.
So the results look like they have the potential impact on those we can get biodiversity analysis.
Right.
So there is also one specific study which seems
like it's impacted already.
It's impacted already. Yeah.
Right. Oh, very good.
So
right.
The results look like logical.
Okay. And then what is your angle on the introduction?
The introduction we were trying to build about the general, the broader aspects of the streams and the relationship between the streams and the bay and things like that. And we were trying to the methodologies.
Right.
What kind of analysis to be done
To investigate.
And we were just going down to what can be done to...
Sounds good. How much literature did you use?
We were going with quite much actually.
Okay.
But,
yeah. Good, because that's exactly that. The story that you're describing there sounds quite useful, and quite constructive. And again, here there's no right answer. There's no right answer. What does, and I said it yesterday, and I'll say it again. What does get a good grade is the good clarity and the logic. And if you can master this, I mean, even in your own thesis, if things die, if it goes to hell, if something, you'll still be able to write your way out of that problem. You'll still be able to write and show that you're a good scientist, even if your experiment goes to heck. Because it's the logic and the clarity. And now there's no word count that matters. There's no number of pages. There's no number of figures. It's act like a scientist. Look at your data. Criticize your own data. Where is it wrong? Where is it possibly weaker? Where is it possibly stronger? What is the aspect of this that actually means something? And that's why the introduction also gives you that background. So it means something. Janova, how about your guys' site?
We don't really have much past papers to work on. We haven't been in BIO300 groups working on at all. So
No, but
the last report is from 2001, and it's just mentioned in a bigger report from Bergen Kommune.
yeah,
We don't really have much to work with, other than that Bergen Kommune said they wanted water quality to be really good by 2015.
right,
It's the last 2001 that they didn't do anything to.
right.
And it's 3-4 times as bad.
Right, interesting. Okay, but even though that particular body of water hasn't been worked on, other bodies of water all over the world have been worked on and have been rehabilitated and investigated. So that's where the literature counts. Previous reports, they'll give you a foot up, but they won't give you a good grade. Really use the literature, the scientific literature and the reports. Wikipedia barely counts, okay? And websites, I'm not so sure. The real literature, use that, because those lakes, those rivers, that's a common problem all around the world. And there's been plenty that's been written about it. because it's possible to rehabilitate impacted waters. It's possible to cut our waste. It's possible to make our water use sustainable. It's possible to make our own waterways healthy enough to in some cases, drink out of without any problem whatsoever. And that's ultimately what we want. But Norway is not unique, your water is not unique, and the methodology is not unique. There's lots of literature about this. And when you start doing the keywords, it's easy. For example, how many references do you have in the introduction now that are not BIO300 and BIO300?
10, 12.
Okay, sounds decent. Hope it is. Right, okay. And how about you, Prakash? How many references?
15 to 20 maybe.
Okay. Good. Olina?
20 of it.
All right. And all used in such a way that you're happy about it?
Yeah,
Yeah.
I think so. We don't know yet.
Right. Okay. Sissa, how about you guys?
We have about 3 pages.
3 pages?
Yeah.
Mhm. Of references?
Yes.
Okay. And in the introduction?
Yeah, the introduction is in 6 pages.
Right. But the number of references?
15.
Okay. Yeah. Good. Yeah, it's coming. Good. And how about you guys?
Around 15 more.
Right. And you? You're with them?
No, I can't remember the number.
Okay. But about the same. Okay. And you? You're with them? Yeah. Okay. So, you've at least got the idea. And the other thing about running through the, see if this works. Yes, it works. Let's see. Okay, you see that diagram at the background. General background for the field of research, the specific background for your problem, the specific problem, including the hypotheses, and a brief summary over the methodological solution. That's it. That is a logical structure to an introduction. Is your introduction logically structured? Right? Okay. The second line of the next last paragraph is revise it often as you change your point of view. Right? And it does happen. Your introduction will have to be revised so that it hangs together with your discussion. So even though you're handing something into the TAs today, and I hope it's in a logical structure and all that stuff and all the references are formally correct, you'll be changing your mind as you build the discussion and as you see that some of the threads that were hanging from the introduction are not actually being picked up in the discussion. And there's only, basically you will see what you take up in the introduction. And the reader will react when new things come up in discussion. But because we're doing it chapter by chapter, you're the ones who know best your own red threads all the way through until it hits me with the final version. Right? And I really do read every bloody little number and comma that you submit, as you've probably figured out by now. And it's just okay. Where has this gone? What has been mentioned? You know, what has been picked up? What has been overlooked? 1 year, for example, extremely excessive phosphorus values were completely overlooked. 1 year, for example, the biodiversity went this way and the text went that way. So when I'm reacting, for example, already to things like a statement that says something is significantly different, and I look at the figure and I don't see how that could be possible, I question it already. because those significant differences are elements of the story that you're building already. And they stick in your mind. Those sentences that you write often stick in your mind better than the actual data. Because those sentences are supposed to have digested things. And they guide the way that you start to approach the discussion and the story that you're building. And that's why I really, really must emphasize, look at the data. As a biologist, and as people on the front line, literally the front line, what you want to do is be able to discover things about the world. Not repeat something that's already in a textbook. You're discovering. And when you discover, it means that some things don't fit the model. Some things will never fit the model because the model is wrong. And it does mean that you have to make sure that your data is good, your data is new, your data shows something else. And that's why it's really important. If your data is good, then the story that's in there is correct, and you can work it as a biologist. There are so many things that we don't understand in biological systems. So many, it's just insane. And then you have to follow the data. Make your data logical, logically put it together, follow the data. A little while ago, I was telling you about when we were doing the how somebody, oh yeah, Vega asked me if I'd ever, had some kind of problem with contradicting previous studies and so on, and that was about the halibut early life history, and this is how on the frontier we were. Nobody knew the early life history from egg to juvenile of halibut at all. There was a guess about Pacific halibut, and there was 4 wild halibut larvae ever caught at that point. 3 at one place and 1 at another. And they were, of course, mashed on. They were identifiable, but they were halibut larvae. That's it. And you couldn't tell what depth they were at. You couldn't tell anything. And so when we were trying to rear them, everything we touched, everything we did was brand new. Nobody knew it. And we had to document, make sure it was solid, make sure what we saw was normal, or decide if it was abnormal, plot out the growth rates, because nobody knew that. Nobody knew the highlights of the development and so on. Fortunately, it goes slow, which is great. But it meant we had data in front of us, and we had to describe something completely new. And we had to describe it in such a way that people understood it and applied it right away. That means the data had to be solid, the interpretation had to be solid, and we had to think out of the box. There was no set way to go. We had to read the data. You had to read the data. And now with the stuff that we're doing on the mucosal epithelia, the barrier tissues of skin, gills, and guts, we developed a new method. There's nobody in front of us. We are applying it in a new way, but it makes sense. There's nobody in front of us, and we have to figure this out. We have to build the data step by step. And the story we're telling is one that has not been told before at all. So read your data. Your data is what brings you forward as a scientist, but also your ability to read it correctly. I can give fantastic data to somebody, and if they can't see the connections, they won't get anywhere. But if they can start to learn to read what the data says, like I wrote in that thing yesterday, is one of your sites in an estuary? Maybe that's why it looks different, why it comes out significantly different and so on. It might have significantly different biodiversity. But read your data. And consider what's weak and what's strong in your data, how much you are emphasizing. If there's a deviant value there, how much is that impacting on the story you tell? for example, we had a science article about the bearded goby off of Namibia. It's changed the ecosystem there. There was 20 of us that had our little parts in there. And it was doing something that was really, really new. And among other things, you know, we have the idea here that fish can't live without oxygen. Well, not only was it living quite happily in a place where there was almost no oxygen down on the sediment, there was also hydrogen sulfide in the water, which is a toxin, and it was doing quite well for that too. And it was able to turn off its own metabolism. And then what the heck was it eating? What was it eating? And it was escaping from some of the horse mackerel and so on. But there was big jellyfish in that area that had come in since the sardines had been overfished. Okay, because there was a huge sardine fishery there. There was big jellyfish. And there was tests about the behavior, whether it chose, you know, a chamber with or without a jellyfish. For some species it mattered, but for the bearded goby, didn't really matter. So it wasn't getting there. And then what was it eating? And our part was, I used to do stable carbon isotopes, well, stable isotopes. And our part was looking at what's the stable isotopes of all the animals in this ecosystem. And the signal was really clear. It's getting 50 to 60% of its growth from the jellyfish. As in, it was possibly hiding in the jellyfish, but its growth, its carbon isotope and nitrogen isotope and sulfur isotope signal was such that it uncontrovertibly said, this fish is living off of jellyfish, and it's able to survive anoxia and toxic conditions, and it's able to respond. You know, it can survive in anoxic conditions for 4 hours, and you touch it with a pencil, and it goes, and it's fine again. I mean, this is stuff that doesn't happen in the textbooks. This is stuff that happens when you're out in the field and you're reading the data. You're figuring out what's happening from your data. So it's these are the things that'll make it really interesting. This is also the story that Anna Christina, who's now at Institute of Marine Research, she spent 2 years working that story so it would get cohesive, so it would get condensed, because there was a lot of information that went in there. I'm tempted to ask you if there's any more questions, and do you want to go at 9 o'clock? Otherwise, I can bring up the podcast of the interview about the story, about this science story, so that you understand how this can go. And then we can have a break. So if you talk to yourselves about whether you want to go right after the podcast or whatever, or if you want to keep working on how to put your stuff together. It's either way is okay for me. But you discuss, and I'll bring this up.
Trenger man at se Jan laten på det. How do we look to the other lab?
All the podcast.
I mean the report.
Hur lång är den på?
Det var det.
Jag kan ju se den.
Skulle du svara på det?
Mm?
Du svarar?
Det var?
Du svarar på det? Ja, okej. Vad är du där för då? Vad är du där för?
7:30.
Ja, okej.
Så jag var bra halv 12.
Ja, så må du ha ett litet.
Så burde jeg egentlig vært hjemme om en tur.
Du ser lost ut. Ja, vi trenger ikke alle til å sitte og gå inn, men det er jo nesten ingenting etterpå. praksis da.
Halv to.
Skulle vi, Janne? Skulle vi snakke om denne påkasten? Prosjektet vårt.
Vi kan godt gjøre det.
Ja, du kan gjøre det neste år, og snakke det på mandag, og så tar vi ut Torborg da.
Ja, vi testet den der Facebook. Ja. Men på mandag. På tirsdag.
Nå må vi ta litt Janne nå, og så får vi med Torborg på mandag.
Altså både mandag og tirsdag har jeg tid.
Ja.
Er ikke vi borte?
Jo, men Facebook-chat eller Skype eller noe sånt.
Ja, det var litt igjen.
Sånn da.
Det var det vi drev og pratet om, at vi burde hatt et møte om podcast før vi skal møte Lars. Vi kan jo gjøre det på onsdag, men da var Torborg opptatt til 3 eller noe sånt nå. Men det går an å gjøre det mandag eller tirsdag.
Ja, nå ja.
Eller vi kan snakke om det nå og bare oppdatere Torborg.
Jeg tror kanskje det er lettest da.
Det er det letteste. Men det er bare at Torborg er ikke med.
Neida, men det blir bare... Ja, så vi kan prate om dette til også, etter her.
Ja.
We'll be talking about the podcast later, before we leave.
Okay, I can't find it right now. But I do know what show it was on. It was on this show here, and if you haven't seen it, if you haven't listened to it, I think you really enjoy it, because it's quite a good popular science podcast. It's been going for about 40 years, and it keeps things going. Okay. But what's your decision? Huh? You want to go work? You want to go work? Yeah, I kind of thought so too. Right. Okay. Basically, if you want the hard copies of the results chapters back here, if there's anything you want me to clarify, I brought them here because if you want me to clarify something, I'll do it now. And if there's anything you want to know more about the introduction, come and ask me now. And remember this part, whoops, sorry. Remember that part there. You may also find unexpected answers to problems that you hadn't even anticipated. That's what you can find in your results. Answers to problems you hadn't anticipated, which means you should be taking them up in the discussion to alert the reader that this might be happening. Okay? So really look at your results. Because this is what's going to make you as a scientist, the ability to read data, the ability to understand what you've actually got. Okay. You want to go work? Go ahead. I'm here to answer questions for about another 5-10 minutes. If you want guidance on what you've already written, come on down.
Gå bort till kantina.
And also notice here, the introduction, the first thing to be read, a draft should be written at an early stage, yet be one of the last things to be revised. Okay? Because this is the introduction to your story, and you don't know your story yet. Okay?
Tackers. We go over to the cantina in here. Ja, vi kan. Nej, vi kan bara ta den här. Bara ta den stopp på det.
That's just something you just explained that in the discussion. But I was also thinking about the yeah, I believe that we may when we're writing the discussion, I think we may have to change a bit on the introduction when it comes to literature. All of a sudden we find out that, okay, we should use this literature and have it split in introduction so
I'll stay here for a couple of minutes, then we have to figure this out.
Tog ut allt.
Så sa vi vi gjerne sende inn alt det ja. Introduction var
Right. So just send it again apples to alla teater.
The font letters. I didn't change the writing. I didn't change it.
Evaluate relative to this, the story has been told in the results, and given that to you as soon as possible, early next week for revision.
So from my part,
Okay.
is
If you find out that she's really a slacker, because now you're going to find out. If you find out she's really a slacker, let me know. Yeah. Okay.
Shall we? Let's do this.
Det var bara dessa här historiska grejerna. Men du sa att du hade mycket i diskussionen och den här.
Sammanläggningar med de förra som det här, det är bra för diskussion.
Ja, men en träning kör ju handlar med introduktion.
Nej. Du kan skriva som att tidigare undersökelser har föregått.
Ja.
Men du kan inte gå i detalj i introduktionen.
Nej. Ja, så var det väldigt fint att få sån tillbakemelding för att se mycket som jag skrev ut klart och sådär. Så det har man lite jobba med. Men jag märker ju också att med tidigare rapporter och förstå hur läst det är lyft upp den här fler. Men där kan jag bara ta kontakt med det.
Okej, ja. Det är det.
Ja.
Okej, men annars, det bara präglar att det här inte blir ganska mycket så det är okej.
Ja. Och så det är liksom den här stick-over-grejen, för där är mycket glömt här att det är stick-over som är byggt över allt. Men lektionen tog vi från i fjol. Så jag tänkte, nej okej, det gör inte. Och så glömde man lite att showa att vi såg ju på något ingenting. Så då var man inte med, hade med det som jag säger på det här, det är lite som när man inte känner
Nemlig.
Men er det noe vi kan skrive at vi så
Ja.
litt på den også?
Ja,
Eller
det kan-
eller at vi
Ja.
ikke la merke til noe?
Eller at metoden som dere valgte ikke var egnet for å tegne inn stickerbacks. Det kan være en metodisk feil.
Ja.
Så du har to opsjoner. At dere velger det bort bevisst, eller at den metoden ikke er egnet for å fange stickerbacks, og at dere ikke konkluderer på om tilstedeværelse er.
Men så har vi for eksempel han Larsson, nå har vi ikke, han har ikke kontaktet oss tilbake igjen, eller på igjen, han er liksom la han være Larsson.
Ja.
Ja da, Sondre er innom stadig vekk, men da han har kanskje hatt noen som har sagt at jeg feller og fått noe der, kunne vi ha brukt en sånn type data.
Men bare gå og snakke med ham. Han elsker å kunne
Ja.
Ja.
Man har brukt så mye til
Ja, nei, altså, hvis introduksjonen er innholdet, ting som er nyttig for resten, så er det helt ok. Ja. Hvis det er bra skrevet, helt ok. Ja, men jeg tror hun har veiledet dere bra.
Ja, jeg var kjempeflink til å veilede veldig. Føles ut som det er litt luksus.
Ja.
Ok. Ja, men leverte inn i går, men så...
Ja, ok. Men hva er det meg?
Ja.
Ja, ok. Er det dere som er med Sigrid?
Ja.
Oh, okay, good.